Alright, today we are looking at Unit 3 of Microbiology, Microbial Pathogenesis. There's also some of the end of Unit 2 that might be going back through at the end of this video, depending on how long it takes to go through this. I'm guessing it'll be 2 separate videos. Alright, microorganisms and mechanisms of pathogenesis. So you have the basic step of exposure. You need to get exposed to whatever the thing is. Then it has to adhere to you in some way. This can be adhering to cells using fimbriae, or their capsule layer might be sticky for cell membranes, or it might be actual protein-protein interactions. Then it has to invade the tissue somehow, and then it will colonize and grow, and then there's 3 possible outcomes, and they're interrelated. You can have toxicity, so these organisms actually produce something that is toxic to the body, or it can be the invasiveness, the actual just act of physically being where it is and using the resources there, causes tissue damage and disease and stuff. Different ways of exposure. You can have various forms of contact from person to person. You can have aerosolized droplets. You can have fluid contact, which would be sexual contact, and possibly through like kissing or something like that. Blood contact, which is related in the category of fluid contact. Really, mucosal tissues are the primary site of adherence and initial invasion. I've talked about the various different kinds. Important to note that fungus, various fungi, also go through this process of adhering and then invading. Invasion is pretty much how something spreads, and the more it spreads, usually the more problematic it can be, but that's not like a hard and fast rule. It's just like a general rule of thumb. Alright, entry of the pathogen into the host. Pathogens use various strategies. Important definition: virulence is the relative ability of a pathogen to cause disease. Pathogen is a disease-causing agent of some kind. LD50, for those who don't know, is the dose of something needed to kill half a group of laboratory animals, and you choose the laboratory animal based on which one's the right model. Usually a mouse for viruses, but bacteria, there's a lot more diversity in what they use. Entry of the pathogen into the host, specific adherence. A pathogen must usually gain access to host tissues and multiply before damage can be done. So you need to attach to something. That's pretty self-explanatory. Bacterial viruses that initiate infection often adhere specifically to epithelial cells through various interactions. So basically, epithelial lines everything, so it makes sense that most things bind to epithelia in some way to get inside the body. This is just showing that you can use a capsule instead of protein-protein interactions to adhere. Bacillus anthracis is the specific species that uses the capsule to bind. These look like microvilli or cilia. I'm guessing cilia in the upper respiratory tract, but I have no idea for sure. Not the focus of this. Entry of the pathogen continued. Bacterial adherence can be facilitated by, this is macromolecules not covalently bind, so this would be the capsule slime layer, which is technically still like just polysaccharides that it excretes like it would in a capsule, so I think these are more similar than he's letting on. And then fimbriae and pili, whatever you want to call them. Fimbriae are the little things that stick out that allow adherence, and then pili can be used for various things like conjugation, which we may or may not talk about. Direct immunofluorescence, so some things fluoresce. Not sure what the purpose of including this is. Those are fimbriae. Alright, so actual entry into the host. Continued. Pathogen invasion. So this is moving to that second part of the actual invasion. And it'll start at the site of adherence, because of course you have to adhere first, and then you invade from wherever that site is. And then you may spread through the host via the circulatory or lymphatic systems. Circulatory, that's your blood going from your heart places, and then lymphatic is essentially blood and fluids coming back. It's an overly simplistic explanation, but it'll work for this. Availability of nutrients is most important in affecting. So the bacteria are looking for something to eat, or microbes are looking for something to eat, and that will determine where they can grow. And so if it's all over the body, they'll spread throughout the body. If it's only in one site, they'll stay at the one site. Virulence, talked about LD50 already. Highly virulent means that there's only a small difference between LD50 and LD100. So if you need like 50 milligrams to kill for LD50 and you need 51 for LD100, then that would be considered a highly virulent pathogen. Whereas if the LD50 of 1 was 50 milligrams and then the LD100 was, I don't know, 600 milligrams, that would not be considered a highly virulent pathogen. Those are, of course, made up numbers. Alright, so this is Yersinia pestis, which is glowing. I'm hoping that it's glowing for a reason. Maybe they put GFP in it, not sure. This is the cause of bubonic plague. Gram-negative, as you might expect. Bacillus, they do have autofluorescence. Okay, so that's their own national fluorescence, not a GFP molecule they put inside of it. It has a structure that helps it kill infected cells. It's also highly motile, and it has virulence factors, so this structure that helps it kill cells is probably its virulence factor. Signs and symptoms: swelling of the lymph nodes, bubones. So this is, you essentially get a bunch of swellings around your whole body. Probably the red marks that were referred to, and these are lymphatic cells. So your lymph nodes get swollen because it's invading all of these lymphatic cells. Spread through an insect vector. These mite ticks, and it eats a blood meal that's not clean, and then defecates there, and then scratching the itch spreads it more. Vector is something that transmits a disease, and vectors live on. The vector lives on the animal reservoir of rats. So rats can act as an animal reservoir for Yersinia pestis. Though he was not, my professor that is, was not very willing to talk about the fact that there's just been a recent study published that makes it seem like it's possible the fleas were not responsible for carrying it around, but for this test I'm supposed to ignore that new information, because that's how things work sometimes. There's a lot of myth and superstition about it, and that God sent it. And you can't really blame them because they had no idea of germ theory, and that wasn't really a thing until much later. Alright, so microbial virulence. A highly virulent organism like Streptococcus pneumoniae. You can see here, this is percentage of mice killed, and this is number of cells injected per mouse, so that can be thought of as dosage for injecting these bacteria. You can see there's a lot less needed of Streptococcus pneumoniae to kill the mouse, and this is about your 50%, and it's very close to the 100% killing. It's not even a full order of magnitude greater. This is a log scale at the bottom. Very important to remember. So this is probably somewhere around 30 for LD50, and then, I don't know, 80 for 90, and probably right at 100, right above 100 for this. Whereas here's your LD50, and you have to go up 3 orders of magnitude to get your LD100. Interesting that Salmonella typhimurium is called less virulent than Streptococcus pneumoniae, especially since Salmonella infections tend to cause more disease in humans. Whereas pneumonia in immunocompetent individuals tends to not be life-threatening. Virulence. Attenuation is the decrease or loss of virulence. Remember, virulence is the ability of a pathogen to cause disease. Right. Go back to this. Relative ability of pathogen to cause disease. So attenuation is the decrease or loss of virulence. This is like when you culture viruses for vaccination. This is basically what happens with some of them, like the measles virus. They have an attenuated strain that's not really capable of causing disease. Toxicity, ability of an organism to cause disease by means of a preformed toxin that inhibits host cell function or kills host cells. So this is a little bit different definition of toxicity than the earlier one. But essentially, it boils down to, does the organism produce a toxin that somehow messes with the host? And can this travel beyond the pathogen? Invasiveness, ability of a pathogen to grow in host tissue at densities that inhibit host function. So that's just, instead of producing any type of toxin, you're growing in places you shouldn't. Kind of the way a lot of the disease phenotypes from cancer work. Virulence. Many pathogens use a combination of toxin and invasiveness and other virulence factors to enhance pathogenicity. So now this is Salmonella. And it's Salmonella enterica, and it has flagella or a flagellum and fimbriae, and it can have virulence factors. It actively releases enterotoxin, and this causes diarrhea. So this is the disease you get if it has the virulence factor. And this capsule around it is meant to mess with the complement system of the immune system. Essentially, they have a bunch of anti-phagocytic stuff on them. So they attack things from a couple of angles. The first is this capsule protects them from macrophages because they can't be phagocytized. That's what this O antigen is doing. And then the enterotoxin is a virulence factor that's produced that will clean out your intestines, basically, causing diarrhea. And I don't know if Salmonella is the one that works by killing your normal cells. I'd have to look. I don't think we need to know. Oh, also important to note, it has an injectosome, which means it can trade this, or not trade, it can propagate this plasmid to other bacteria. Blood agar plates, you look for clearing areas. Alright, virulence factors. Pathogens produce enzymes that enhance virulence by breaking down or altering host tissue to provide access to nutrients. Hyaluronidase, I assume that's part of UTIs. So you literally alter host tissues to get something nice out of it, or you can protect. So you can interfere, or you can break down, essentially eat part of the host, or you can try and evade the immune system. Exotoxins. These are proteins released from pathogen cells as they grow, and they come in three basic categories. And I'm probably going to have to know these. So cytolytic toxins, AB toxins, and super antigen toxins. Cytolytic toxins, hazard guess here, they will just lyse the cell. AB toxins, I don't quite remember. And then super antigen toxins probably cause cytokine storm, that kind of stuff. I don't know. Let's look. Cytolytic works by degrading cytoplasmic membrane integrity, causing cell lysis and death. These toxins are called hemolysins. So, yeah, pretty much what I thought. And so this is a blood agar plate that's got clearing zones around Pyogenes. If it's completely clear, then it's positive. Oh, this is one of these cytolytic toxins. You have alpha toxin. And the alpha toxins essentially are shipped out as it's an exchanger with various extracellular things. AB toxins consist of A and B. Oh, B binds to receptor, and then A does something damaging to the membrane. And tetanus and botulinum, diphtheria all do this. So binding is B. That makes sense. Bind is for B and A is for activity. That actually makes quite a bit of sense. So we have cytolytic and these AB toxins. And the action of... So this is diphtheria toxin. Let's go back to it. And so it's not on this side. Here's some amino acids going through a ribosome with the tRNA coming in, and you have to up and out. But then if you have diphtheria toxin, first B binds to this receptor, A gets internalized, it has its activity which messes with EF2, which means you can't get your next tRNA in, so you won't translate the protein. And then this is botulinum toxin. This just shows acetylcholine being put onto a muscle at a neuromuscular junction, and then this is if you have botulinum here, it blocks the release of this acetylcholine. Again, it's binding with the B domain, and then the A domain is somehow inhibiting the other functions, probably affecting synaptobrevin or snap or snare proteins. And this is tetanus, which ends with the same result as the botulinum, right? And that it will cause no, it'll cause the opposite. Tetanus toxin blocks the inhibition of the neuromuscular junction, which causes it to become overactivated. And that means that these inhibitory glycines aren't fired, and these are always firing, which will cause you to tense up. So tetanus toxin, not a fun way to go. And then superantigen toxins worked by stimulating large number of immune cells, results in inflammation and tissue damage. So not quite a cytokine storm like I was thinking, but it's pretty close in that you just want to essentially convince the immune system to overreact and damage the surrounding tissues. And then enterotoxins, these are a special type of exotoxin, I would think. But maybe they're a completely different category. Exotoxins, okay, so they're in the same category, whose activity affects the small intestine. So entero essentially stands for gut, and your small intestine is the longest part of your gut. Generally causes massive secretion of fluid into the intestinal lumen, resulting in vomiting and diarrhea, like cholera. Okay, that's pretty straightforward. Cholera messes with your first, it's from the AB exotoxins, but yeah, it essentially just messes with the ability to produce camp. Well, the actual blockage blocks sodium from moving in, and so Cl must move out to the lumen, as does bicarbonate, and that causes water to follow them, and that follows them into the intestine, causes this wastewater stool, and the actual receptor is the CFTR, which is what's mutated in cystic fibrosis. So it's interesting. So if you're heterozygous for a defective CFTR, it will actually protect slightly against this cholera effect, cholera toxin. Endotoxins, like the LPS portion of the cell envelope of gram-negative bacteria, and they're generally less toxic than exotoxins, which is good news for us. Host risk factors for infection. Compromised host, one in whom one or more resistance mechanisms are inactive and in whom the probability of infection is increased. Age is an important factor for this, so as you get essentially beyond 30, your immune system starts going downhill. But that usually doesn't show up till late 50s, early 60s, and then it gets steadily worse. Stress can predispose a healthy individual to disease, because stress will downregulate your immune system, because cortisol, and diet plays an essential role in host susceptibility. Your body's only as good as what you put into it, so if you eat all crap food, you won't have a very good immune system because it can't build itself right. Certain genetic conditions can compromise those. You can't really do anything about those. Innate resistance. So natural host resistance, tissue specificity, physical and chemical barriers. This is the part that doesn't have memory, it's just your innate resistance. So your eyes bathed in fluid that has lysozyme, your skin produces sweat, and it has sebaceous glands which produce sebum, and it also has lactic acid which keeps it mildly acidic, like 5 to 6 pH. Movement of air through the lungs and streaming of urine are meant to stop stuff from being in it. The ciliary elevator in the lungs pushes the mucus up and gets rid of the phlegm and stuff that traps microbes. And the liver makes various antimicrobial proteins. Obviously, the pH of the stomach prevents bacteria from growing there. And the specific tissue is important. So, natural host resistance with that first slide. Specific tissue is this slide. So, it's like for HIV infection, you need to infect T helper lymphocytes. It doesn't really infect the other cells. It does infect some other cells, I should say. I shouldn't say it doesn't infect anything else. Microbes must always have countermeasures to the body's resistance mechanisms. An example is diphtheria, which has a poly and structures that help it stay attached to the throat and not moved out. Okay, this is Chlamydia trachomatis, which is an obligate intracellular parasite, and it's gram-negative based on sequence similarity. It causes these weird bumpy patches on the eye, and that'll eventually on the eyelid, and that'll eventually cause you to scratch your cornea, and that's no good. It's one of the most common STIs, STDs, whatever you want to call them, and it goes on both types of mucosal tissue, so the eye and in genitalia. And there's dozens of serobars, which is quite a bit different from Salmonella that we talked about earlier that has 2,000 serobars, something like that. This is treated by antibiotics, and PCR-based test will identify it because it is not growable in laboratory culture. Isolation of pathogens from clinical specimens. If a physician suspects a disease is caused by an infectious agent, samples of tissues or fluids are collected for microbiological, immunological, and molecular biological analyses. Samples may include urine, blood, urine, feces, sputum, cerebrospinal fluid, or pus. So, you want what you would expect things to be, where you would expect them to be. Lab ID of unknowns, there's a bunch of things you can do. Important to note, ELISAs are antibodies that are directed at proteins from specific pathogens, so HIV tests are done with ELISAs. And you can also look for other antibodies, so the antibodies in a patient's serum. You can also use molecular techniques like PCR to detect things. So this is just normal throat swab. Isolation of pathogens from clinical specimens continued. Tentative identification of an isolate is often achieved by observing colony characteristics on various media. And then you do more detailed tests, which is what we're doing right now, various biochemical analyses to figure out an unknown. Colony characterization of frequently isolated pathogens. They're not important right now. Some pathogens are easily identifiable under a microscope, and Neisseria gonorrhea is one of them. And this is why, is it forms these nice little intracellular patches. So this is a cell, and then the little black things inside are Neisseria gonorrhea. And it's really one of the only things that looks like that. It grows inside white blood cells and is gram-negative, which you would expect. And then you can use the Thayer-Martin media, which is selective for Neisseria, and a coloring agent will tell you if it is gonorrhea. So if it grows on this Thayer-Martin media, it will be Neisseria. It's selective for Neisseria. Growth-dependent IDs. Pathogens are often subjected to biochemical tests, like I said, we're doing right now. There's specialized media that are selective and differential, and then different media incorporate these biochemical tests. I thought this would be the picture. So this is clinical diagnostics, and these are just little notes about various things. Decarboxylation says that something is enteric, but otherwise these are not that interesting. Catalase is good for staph versus strep. This is on this one. Staph would be positive. This is growth-dependent diagnostic media. If they produce acid, they turn yellow like this, and then they produced gas, the little Durham tube will go up. This one looks like it's suspended a little way above the bottom. Enterobacter, you would expect to produce a lot of gas. This is actually more basic. This is the control. This is actually more basic because it's become redder. We're not doing these really. This is the citrate test. It'll turn green. Pretty straightforward. So these allow you to do a bunch of biochemical tests at one time by putting a syringe through them. In the real world, this is an automated process. Disk diffusion. I need to do this yet. This is a standard procedure for assessing antimicrobial activity. It's used to determine an organism's susceptibility to an antimicrobial agent. So you put basically antibiotics and you measure the clearing zone around them later on a plate. So you spread them out on the plate, and then this is after everything is said and done. In the center of each of those circles is a antibiotic of some kind. The clearing zone tells you relatively how well the process actually worked. And you measure the diameter of these zones of inhibition. And then antibiotic susceptibility. You can have a dilution method, and it's usually by an order of magnitude, but you can do any dilution you want. And then, like, this would be the minimum inhibitory concentration, because it's the smallest concentration in which you have inhibited all growth. This is the E-test, which is similar, except you have these basically ruler things there already. And it's on a gradient. And the gradient is known, and so wherever this stops, you know the concentration. That's the minimum inhibitory concentration. Instead of the minimum inhibitory concentration. Okay, it's both concentration. The MIC. This is Penicillium chrysogenum, and it produces penicillin, normally. Not the kind we actually take. Sorry, it's raining out today, and apparently that's making me yawn quite a bit. This is a fungus, and the penicillin it produces is not as potent as the semi-synthetic one we have. Semi-synthetic, if you hear that word, refers to something we found in nature, usually an organic molecule, that we have added our own designs to make it more potent, less potent, more efficacious, less efficacious. And penicillin is one of the best examples of a semi-synthetic antibiotic. This was discovered by Fleming. He had been culturing staph, and then some had mold in it, and he eventually found this instead. Alright, this is Streptomyces griseus. It produces over 6 antimicrobial antibiotics. Some have been used in situations as anti-cancer drugs, herbicides, and more. 90% of useful antibiotics are semi-synthetic, so you find something in nature like this, and then you modify it with organic chemistry to get a very powerful product. The original penicillin was only active if injected. Now, amoxicillin can be orally taken. Another thing is Streptomyces hygroscopicus produces an antibiotic of some kind, and it's strange in that it's produced by a bacteria, streptomycin, not by fungi. There's also an immunosuppressant agent that it produces. Derivatives are used in clinics. Herbicides and parasitic. Alright, so here's some mechanisms of action. These I'm going to have to know. So tetracycline, which works by, I'm going to guess, oh, I have no idea. Protein synthesis. Okay, so tetra means 4. Tetraprotein synth inhibits protein synthesis on the 30S subunit. Tetra. I don't want 4 of that protein. I want none. Tetracycline. Streptomycin. This also blocks protein synthesis, doesn't it? Looking for it. Is this streptoviricins? I don't think so. No, here it is. Streptomycin. Yeah, protein synthesis. It's exactly the same as tetracycline. Good to know. And then gentamicin. Gentamicin is again the same thing. By the way, I do not agree with this particular unit of learning some of these, or how we're learning these. I think it should be its own unit to itself if we're going to remember them, but that's just me. Puromycin is over here. Is protein synthesis to tRNA level. Pure T. Gentamicin. For general protein synthesis. These 3 are together. I remember streptomycin, so if I can just remember STG, St. Generous, right? They all have the same function. Alright. Potentamycin, which is a completely different mechanism than everything else, which is the lipid biosynthesis. Which plants need oil. It messes with lipid biosynthesis. Potentamycin. Sure, why not? Trimethoprim is right here. It's folic acid metabolism. Interesting. Mnemonic for this one. Trimeth, that's like 3 months. 3 months, and that would be part of pregnancy, and therefore it's whatever would affect folic acid. Could say 3 trimesters. That would make a lot of sense. 3 trimesters in a pregnancy. Potential acid affecting folic acid. And vancomycin, which is the last one that's in its own category. But it's still just so well synth. As are the remaining 2. Antimicrobial spectrum is the range of species that something can interfere with. C tetracyclines have the broadest spectrum. That's why they were the most abused and now are held in reserve. Vancomycin is very narrow. Gram-positive cell wall synthesis. Okay. Drugs for viruses don't work in bacteria, vice versa. There's a direct correlation between how much antibiotics are used and how much resistance shows up. So, don't overdo it. Animal worldwide production and use of antibiotics. Right now, the cephalosporins are the big thing. Used to be the penicillins. Now they're 17%. Still quite a bit. And tetracyclines have been cut off to ridiculously low levels. And there's some naturally occurring antimicrobial drugs. Antibiotics are naturally produced antimicrobial agents. And less than 1% are clinically useful, though they can be modified by semi-synthetic processes. Susceptibility of microbes varies quite a bit. There are a broad spectrum that affect both species, both gram-negative and positive. So this is beta-lactam antibiotics, so penicillins and cephalosporins. They're going to affect cell wall synthesis, discovered by Alexander Fleming. Alright, and discovered by Alexander Fleming. Obviously going to work more on gram-positive. Nothing particularly interesting. Beta-lactam ring is the square ring. It's very highly reactive, and it breaks up transpeptidase activity, which means the links of the cell wall never get made. The protein links. Different R groups in semi-synthetics are pretty much, so on this one the R group is right here, and by changing that R group you change its ability to cross barriers and get into places and stuff. Diazolidine ring. Oh, and it's got a 5-membered ring, which is different from the cephalosporins, which have a 6-membered ring. Cephalosporins produced by the fungus Cephalosporium. Same mode of action as the penicillins, and they're commonly used to treat gonorrhea. So it's still affecting cell wall synthesis with the beta-lactam ring. It's got 6-member. It's got this dihydrothiazine ring. More antibiotics from prokaryotes. Aminoglycosides contain amino sugars bonded by glycosidyl linkages. So this is your kanamycin, neomycin. Not ampicillin though, because that's in the penicillin family. And they're not commonly used because they cause a lot of toxicity. In both the this is nephrotoxicity, so nephro means kidney, so in your kidneys, and the neurotoxicity, that's your neurons in your brain and stuff. So that'd be bad to lose. Considered reserved antibiotics for when everything else has failed, because the side effects are worse for these than anything else they'll give you, probably. Macrolides, they have a lactone ring bonded to sugars, so instead of being like these aminoglycosides, which are pretty straightforward amino acids with glycosidic linkages, these have lactone rings, which are not explained what they are, bonded to sugars. Lactone, probably like lactose, except bonded to sugars. Erythromycin, one of the more popular antibiotics, is one of these. And the reason is it's very broad spectrum since it targets the ribosome. Tetracyclines are the other broad spectrum, and they're on the other half of the ribosome, or whatever percentage of the ribosome that is. Microbiology inhibits by binding into a large target subunit, prevents translation. Oh, so that lactone ring just since it binds to the large subunit, it stops on translation. Daptomycin, also produced by streptomyces, not just streptomyces, and it's used to treat gram-positive bacterial infections. Forms pores in the membrane that cause depolarization. Well, that could be bad. So, daptomycin is like digging, and you dig and you put in tunnels, and these tunnels let in stuff from the outside. Platentamycin is a new structural class of antibiotic, broad spectrum, and it works against MRSA and VRSA. And platentamycin is the one that does. What does it do? Wasn't this fun? On the graph, various ones. Platentamycin is the lipid biosynthesis. Right. So lipid biosynthesis is a weird thing to mess up. Plants produce oil, and oil is made up of fatty acids and stuff. Lipids. In other words, tetracycline, 4-member ring, can have various armed groups. Yeah, 4 of them. Their positioning is what's important. Daptomycin, which puts a hole in the membrane, or essentially a pore in the membrane to let stuff in and out. This is platentamycin, which affects lipid biosynthesis. Not clear why. So this, you can kind of see this tunnel here. Probably not how it works, but just for memory reasons. And then this, see these are how that works and the diversity you can get out of that. This one, not as clear. It's probably an enzyme that goes with it. Antiviral drugs, they're not very good. There's a few that specifically target a virus. Most commonly used are nucleoside analogs or non-nucleosides, reverse transcriptase inhibitors. So you have nucleoside reverse transcriptase inhibitors, non-nucleoside reverse transcriptase inhibitors. Protease inhibitors stop various proteins. So this is some HIV drug. Part of the HIV therapy does this. Fusion inhibitors prevent viral fusion. And there's two categories of drugs successfully limit influenza infection: adamantinase and neuraminidase inhibitors. So neuraminidase is needed by influenza to enter cells. And then adamantinase, I don't know what that is. Interferons are small proteins that prevent viral multiplication by stimulating antiviral proteins on uninfected cells. There's only a limited number of things we can target because viruses have multiple gene products and then cleave them, and then interferons and cytokines act as an alarm system. And it was thought that you could just add interferon to somebody to feel better, but they'll feel really sick because interferon causes the sick feelings and fevers and stuff. Alright, antifungal drugs. Fungi pose special problems for chemotherapy because they're eukaryotic. Their cell machinery is a lot like humans, so you can't use like streptomycin or daptomycin and that kind of stuff. As a result, many antifungals are just topical, and there are just a few drugs that target actual metabolic things. Ergosterol inhibitors target unique fungal plasma membrane component, ergosterol. So ergosterol is an important part of the membrane, which probably if you get rid of it, it's very important for structure. Can treat candida infections. Here's some antifungals. What was that called? Echinocandins. They have echinocandins that inhibit this, and they affect the membrane in some way. And then ergosterol, that's what you're stopping. Yeah, ergosterol inhibitor. Or antibiotics. That's what you're targeting. Antimicrobial drug resistance. Yes, the acquired ability of a microbe to resist the effects of chemotherapeutic agent, which is normally sensitive. So there's so there might be lack the structures that the thing requires. It might be impermeable. It might be inactivate the antibiotic. Might modify the target. Might develop resistance through a biochemical pathway, or it may just pump out the antibiotic. Oh, it's very good. So might pump out, might not have the structures, might retarget, and might inactivate it somehow. I guess that's just for rate at which antibodies are attacked by enzymes. Not quite sure what I'm supposed to get out of this slide. I guess this is an enzyme that targets that square ring in beta-lactam rings. Yeah, not sure what that's all about. More drug resistance. Most are isolated from patients on R plasmids, and R plasmids predate the antibiotic era, so they've been around for a long time. And the use in agriculture, especially in then medicine and the veterinary offices, explain why the R plasmids are all over the place now. Currently, we overuse them for agriculture and stuff. This is just showing how quickly drug resistance in Neisseria gonorrhea is going. Almost all pathogens have a strain has acquired some resistance. A few have done it to all known microbial agents, and then versa would be part of this. And resistance can be minimized by using antibiotics quickly and only when needed, and resistance to a certain antibiotic can be lost if antibiotic is not used for several years. So if it goes out of the cycle, the things will lose the R plasmid, and then you can start using it again. Alright, not really important here, just know that Staph aureus has really got the cornered the market on drug resistance, but look at all of these species. They search for new drugs, and so this is the focus of a lot of current work. You want to modifying a current agent is easier than making a new one, and this drug discovery process has become ridiculous. So 1 in 7 million drugs are right. So here we have an arm that you'd have like 3 data scientists dedicated to while it goes into these experiments. And it's a huge chemical library. Oh, wow. Alright. Here's the chip that they use for that, so you can do very high throughput. These are incubators for those things. And then computers now can try and predict ways to do this. So making the saquinavir that's part of heart therapy targets the HIV protease. That was a bottom-up design using computers. And then there's natural products. Usually you'll find one platensmycin being used for the various antibiotic resistant things now. Platensmycin, lipid biosynthesis. Combination of drugs is used, so heart for HIV. And bacteriophage therapy is starting to be a thing in Europe, where they're seeing if they can infect the bad things inside of you. Not important for this. AIDS pandemic. We talked about that already. So I'm curious if we're gonna do that again on this time. I mean, AIDS is caused by HIV. There's 25 million people who have died. There's at least 70 million people with the virus. Alright, healthcare associated infections. These are the most common antibiotic resistant infections. So these are called nosocomials. I missed what the actual word was. Something like that is the fancy word. But by far the most common are urinary tract infections, and then surgical infections. These HAIs, they usually have something that wasn't present upon admission. Nosocomial, that's the word I was looking for, not nosocomial. It's close. There are about 1.7 million HAIs per year in the US. So globally it's probably much, much higher. Some are acquired from patients, but most are from some part of the hospital environment. So they're on phone lines of some kind. Many patients have low resistance. They treat infectious people. There's multiple patients in a room, and healthcare personnel move from person to person, and healthcare procedures breach your integumentary system and outside defenses. More about these. Newborns, particularly susceptible. That makes sense. Surgical procedures expose organs to contamination. So we saw this was the second most likely cause. Go back to this chart. Surgical infections are the second most likely, and that makes sense because you're being exposed. Newborns don't have an immune system yet, a challenged immune system yet. Certain drugs will increase the patient's risk, especially like chemotherapy drugs. Use of antibiotics is selected for antibiotic-resistant organisms. So just the sheer weight of the amount of antibiotic they use in a hospital probably creates some of these strains. This is just about nosocomial infections. Public health measures for disease control. So we look at the reservoir and see if we can eradicate the reservoir. If not, that's sad. It's easiest if the reservoir is humans to eradicate something. That's why smallpox was the first thing eliminated and why polio is the current target, because there's no animal reservoir. We are the reservoir. Immunization is a great, wonderful health measure. We've eradicated smallpox because of it. Measles, mumps, and rubella have been on their way out, though the resurgence of anti-vaccination campaigns has meant that they have not yet been closed out, but they have animal reservoirs. So. Well, I know smallpox has cowpox, I'm not sure about the others. Quarantine, so restricting movement, surveillance. You essentially survey things to find disease, especially the data from the H1N1 in 2009. We've actually got pretty good surveillance in place now. And then pathogen eradication, so you want to remove the whole thing, and then these are useless slides. Alright, that will be enough for today's video, and perhaps I will do the final part of Unit 2, just so I can have that done as well. Alright, thank you. If you want to reach me, you can reach me at krebradio@gmail.com. That's krebradio@gmail.com.